Here we are again with our botany and dear students of 12th class, and today we are going to take up one of the most important topics in this genetics, that is the transcription. After in the replication of the DNA, we are going to discuss or take on the transcription. Now, the process of copying genetic information from one strand of the DNA into RNA. Transcription is the process of copying genetic information from one strand of the DNA into RNA. Also, the principle of complementarity governs the process of transcription. Here, there is only one exception, that the adenosine now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which one certain, that means when the process of replication starts and nick after the nick, the total DNA of an organism gets duplicated, and it does not stop. It continues till the whole DNA gets duplicated or replicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. And this necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. That means at a particular juncture or at a particular time, as per the need of the organism or the body of the organism, a particular part of the DNA gets transcribed into the RNA. And now this necessitates defining the boundaries that would demarcate the region and the strand of the DNA that would be transcribed. And now here, why both strands are not copied during the transcription has the simple answer. First, if both strands act as the template, they would code for the RNA molecule with different sequences. And in turn, if they code for proteins, the sequence of the amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. And second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. And this would prevent RNA from being translated into protein, and the exercise of transcription would become futile or useless. And now, what defines a transcription unit? The transcription unit in the DNA is defined primarily by three regions in the DNA. First one is the promoter, second the structural gene, and the terminator. And there is a convention of defining the two strands of the DNA in the structural gene of a transcriptional unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase, it is DNA-dependent RNA polymerase, it also catalyzes the polymerization in one direction, that is 5'3'. The strand that has the polarity 3'5' acts as a template and also referred to as template strand. The other strand, which has the polarity of 5'3' and the sequence same as the RNA except thymine at the place of uracil, is displaced during transcription. Strangely, the strand which does not code for anything is referred to as the coding strand, as the transcript produced is having the information coded just like the base pairs of this coding strand, and all the reference points while defining a transcription unit is made with the coding strand. Here is the hypothetical sequence from a transcription unit. On the upper side is a template strand, and below lies the coding strand. And easily we can show the transcript that gets formed from this template strand, and the base pairing is similar to that of coding strand, except that in place of thymine there is uracil in the RNA transcript that gets formed. On the left hand side, this is the structure of the transcription unit. On the left hand side is the promoter site and then the structural gene, and on the right hand side is the terminator or the terminating end of the particular DNA strand. And now the promoter and the terminator flank the structural gene, as you can see above. In a transcription unit, the promoter is said to be located toward the 5' end upstream of the structural gene. The reference is made with respect to the polarity of the coding strand. If a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and the coding strands, and by switching its position with terminator, the definition of a coding and the template strands could be reversed. And the terminator is located toward the 3' end downstream of the coding strand, and is usually defined the end of the process of transcription. And there are additional regulatory sequences that may be present further upstream or downstream to the promoter, and some of the properties of these sequences we will discuss in the forthcoming chapters, inshallah. And this is all about transcription, and for its detail that is to be studied in the higher classes. Here, as per the requirement, how the transcription unit gets formed from a particular DNA strand is defined in a brief manner that is required at this standard that is at your 12th level. Thanks. Have a nice day.